Hello, everyone. Esper, thank you again for inviting me to this wonderful occasion. Oh, there's still music. That's nice. I think it's appropriate, actually. You know, the science is excellent. The company is excellent. Apart from Rick, the food is wonderful. It's a whole package, and I look forward to meeting the students and the others for lunch at 12 in the Renaissance Hotel. So thank you. So this is a whole package. My talk is not a whole package. It's basically a work in progress. But I thought it got to a stage which is interesting enough to show people. And it's called Predicting Who Does and Who Doesn't Become Infected. So this story started a few years ago when the Vaccine Research Center, we have a scientific advisory board every now and again, and our labs are interrogated by experts. And it was about the time that the vaccine trial called HVTN 505, the results had just been released. And the trial of this vaccine to prevent HIV was not successful. And I was giving my talk about other things. And the head of this committee asked me, I think unfairly, why I thought the vaccine didn't work. And I said, I have no idea why the vaccine didn't work. I don't know. I didn't know why some people got infected and some people didn't get infected. And then I thought about this, and I thought, you know, we really don't know why, when all things are equal, okay, in similar risk settings, why some people become infected and some people remain uninfected with HIV. Now, we know that there are a number of factors that affect and effect HIV acquisition. There are geographical factors, such as HIV prevalence. The more HIV there is, the more likely you are to get it. There are behavioral factors, such as the number and frequency of different sexual contacts. And there's the nature of that sexual contact itself, whether the contact is protected or unprotected, whether you're circumcised or not, for example. And there are biological factors that we know about, such as host genetics, CCR5. If you are a CCR5 delta 32 homozygous person, generally speaking, you don't get the infection. But there aren't many biological factors such as that we've identified. The virus load in the transmitter is very important. If it's higher, you're more likely to acquire the virus. And we know that sexually transmitted infections are important. If you have, for example, herpes simplex virus 2 infection in the genital tract, you are at much greater risk of acquisition of HIV after exposure. So in my world, we deal a lot with inflammation. And inflammation is bad for you at many points in the disease, but especially during acquisition, which is what I'm talking about today. There's a lot of data suggesting that immune activation and inflammation predispose to HIV infection. And as I just mentioned, if you have sexually transmitted diseases with inflammation, that increases the likelihood of transmission and also the number of transmitted founder viruses. And there are a number of other factors which link heightened inflammation and HIV acquisition. However, for every study showing a link between heightened inflammation and acquisition of HIV infection, there's another study suggesting that immune activation and inflammation, because of activation of innate immune pathways, actually protect against HIV infection. So it's very much an open question. So when in thinking about this, I thought, well, that the HVTN 505 vaccine trial would be a great setting in which to ask this question. All things being equal, why do some people become infected and some people not? So let me just remind you about the HVTN 505 vaccine trial. It was a phase 2b randomized placebo-controlled test of concept trial, and its aim was to evaluate the safety and the efficacy of a multi-clade DNA prime recombinant adenovirus 5 boost vaccine. The people who were recruited for this study were HIV uninfected, obviously. They were antibody negative against AD5, and they were all circumcised, men who have sex with men, and male to female transgender people. The study enrolled about 2,200 people across the United States. And you can see what the vaccine was like here. At month 0, 1, and 2, a DNA plasmid was given which encoded ENV, GAG, POL, and NEF. And then at month 6, a recombinant AD5 virus vector was given, encoding ENV, GAG, and POL. And the primary endpoints were as follows: HIV infection after week 28 following enrollment, so it's after this time point, through the 24-month of the study, and HIV load at set point if you became infected. And there were visits at these months here up to month 9, and then every 3 months through 2 years. So HVTN 505. It was stopped for futility. As you can see here, there was no significant difference in infection events between the vaccine group in red and the placebo group in black. Some vaccine and some placebo recipients became infected. Some vaccine and some placebo recipients remained uninfected. So in similar risk settings, high risk, men who have sex with men, why did some of them become infected with HIV and others not? So what we did, from the 2,200 people, we got 83 participants who were selected because the cases were controlled with the uninfected people. So very controlled. They were matched for age, ethnicity, risk factors, a whole bunch of other things. We had 39 cases of people who became infected and 44 controls who remained uninfected. 47 of those had been vaccinated and 36 of those had received the placebo. And we got samples from 3 time points: baseline pre-vaccination, first sample after the recombinant AD5 boost immunization, and month 12, 6 months after the boost immunization. All the samples that we got to study, I'd like you to remember, were all pre-infection, all right? So we're looking for something pre-infection that will allow us to predict who becomes infected later on. We got plasma and we got PBMC samples. So something that's very important, and we'll look at this briefly and then put it aside, the study group characteristics. So we're comparing the whole trial cohort to our 83 people in our study cohort, and there was no significant difference in clinical characteristics between our study group and the overall trial cohort, except, of course, for the fact that we had 39 HIV-positive people, which is nearly half of our cohort, whereas it was only 2% of the whole trial cohort. Apart from that, the clinical characteristics of our cohort reflect those of the overall trial cohort. So, we have a kind of vague question: why do some people get infected, others not? We don't know what the answer is. We don't really know what to do. So, in this situation, we just measure everything and hope for the best. We call that multifaceted analysis of immune status. Some other people call it systems biology, and yet other people, in a rather disparaging fashion, just call it a fishing expedition. So, going fishing is not such a bad thing, as you'll see later. So this is me fishing. This is my line, and somewhere in there, there's the fish. Now, I can't see the fish, but I do know that he's in there somewhere. So, multifaceted analysis of immune status. We took plasma, and we did ELISA, Luminex, and PCR to measure a whole bunch of soluble analytes. We did flow cytometry to look at the phenotypic status of a whole bunch of immune cells in peripheral blood. And we took those cells, and according to certain phenotypes, we sorted them by flow cytometry, and we did whole transcriptome sequencing, RNA-seq, to look at the gene expression profiles. So first of all, the soluble plasma biomarkers. So there they are at the top. There's I don't know how many, like 30 of them. Most of them are markers of inflammation, immune activation, stuff like that. I'm showing you a few of the important ones here that we know are terribly important for HIV disease progression, such as D-dimer, soluble CD14, IFAP, IL-6. We found no significant difference in soluble plasma biomarkers between the uninfected controls and the infected cases at or across any visit. And we couldn't construct a computational model that combined these plasma biomarkers into something that could predict HIV acquisition. So the soluble plasma biomarkers were of no help. So what about surface phenotypes of the subsets in blood? So we used a 16-color phenotyping panel. These are the markers that we used here to look at all the different phenotypes of various cell subsets. And 7 of the cell subsets, these ones here, we also sorted them according to these phenotypic markers, and we performed transcriptomic analysis. I'm going to talk about that later. Let's look at the phenotypes first of all. So, very importantly, we found no differences between the vaccine and the placebo recipients in terms of the different phenotypes of T cells and the other cells that we looked at. However, if we looked at the frequencies of activated memory T cell subsets, we found that those could differentiate between the uninfected controls, you can see those in blue here, and the infected cases in this principal component analysis. And what you can see, if it's in red, it means that this is a higher frequency in the infected cases. If it's in blue, it's higher frequency in the people who remained uninfected. There's a whole bunch of markers that are higher and can differentiate significantly between those who became infected and those who remained uninfected. Each one is significant on its own, but in a multivariate analysis, only the HLA-DR positive activated CD4 T cell subset remains significant. So we've got a signal that can differentiate between becoming infected and remaining uninfected. So what we want to do now is to build a model to predict acquisition. So let me go through how we build this model of acquisition. Say we have a bunch of markers here, 1, 2, 3, 4, 5, 6, 7 different markers, and they correlate with becoming or not becoming infected. These markers up here, like marker A, correlates very well with becoming or not becoming infected. Marker G, and these ones down here, correlate very poorly because their correlation coefficient is low. What we want to do is to build a model such that each combination of markers gives a variable of prediction with a different accuracy. So we want to be able to predict the controls as controls and predict the cases as cases. Now, if we base our model on these markers with a higher correlation coefficient, we have a pretty good shot at predicting the cases as cases and the controls as controls. If, however, we use the markers with a low correlation coefficient, we have a very poor model, right? The controls are predicted as cases, and the cases are predicted as controls. So multiple combinations or variables are used to build this model. And if we use all of these markers, we have a pretty crappy model of prediction with an accuracy of only 20%. So what we'll do instead is to use the markers that have high correlation coefficient that you can see there. And if we use all of those, we can use them to build a model with a high accuracy of predicting cases as cases and controls as controls. So the more markers with strong correlation give a model with higher accuracy of prediction. And so what we did, and this work was carried out in Rafik Sekali's group by Slim Ferrati, really nice piece of work. What I'm showing you here is the markers that were used for the prediction model at month 0 and the markers that were used for the prediction model at month 7. If it's blue, there's a higher subset frequency in the controls who did not become infected. If it's red, there's a higher subset frequency in the cases who became infected. And the regression coefficient, as I just said to you, gives the relative contribution of the variable to the prediction. So we can see a few things here. First of all, quite importantly, the markers that make up this model at month 0 are pretty much the same as the markers that make up the prediction model at month 7, all right? There's a state here which you can use for prediction at both month 0 and month 7. That tells you something important about the prediction. Also, there's a contribution from both innate, you can see here activated monocytes and NK cells here, and adaptive immune components such as the frequency of CCR5 positive CD4 T cells. So if we put all of those markers together and we build a predictive model, what we do is to take the 83 participants in our sub-study, we randomly split them into a training group and a test group. Both groups have similar characteristics, and we build the model. And what we found was that prediction models based on cell subset frequencies at month 0 or month 7 are able to most accurately separate HIV cases from HIV controls with an accuracy of about 65 to 70%. So we can predict who's going to get infected with a reasonable accuracy. It's not great, but we're getting somewhere. And remember, it's based on inflammation and immune activation. The more of that you have, the worse you are off in terms of acquisition. So remember, we then did whole transcriptome sequencing on those cell subsets to look for gene expression profiles. And we looked at these subsets here: NK cells, monocytes, myeloid dendritic cells, B cells, and T cells. We looked at CD4 memory, CCR5 positive and CCR5 negative T cells, and CD8 memory T cells as well. And with the whole transcriptome data, we can do a bunch of different things. We can look at differential expression of individual genes. We can look at the expression of whole pathways. And we can look at the sequences, insertions, deletions, SNPs, and alternative splicing. So the first thing we did was to assess whether there was a significant difference in gene expression between the vaccine recipients and the placebo recipients. How we did it is a bit complicated. To be honest, I don't really understand how, but I just like saying this word here, Benjamini-Hochberg correction. It sounds wonderful. I recommend when you're on your own, just say those words to you. They're fabulous. So we employ the Benjamini-Hochberg correction to look for evidence of difference in gene expression, and we found none. So the vaccine and the placebo groups, in those cell subsets, there's no difference in gene expression, all right? Very important to establish that first. So now we look at the gene expression of individual genes, and we look at the pathways. So we're going to look, first of all, for significantly differently expressed genes between cases and controls. We're looking in these subsets here. The controls are in blue, the cases are in red. So if there are differentially expressed genes which are higher in the controls, they're shown in blue. If they're higher in the cases, they're shown in red. And what you can see here is there are some genes that are differentially expressed between the controls and the cases, but not many. Okay, not many. So let's look at the pathways now, the gene expression pathways. Now what you can see here is that there are many significantly differently engaged pathways between the cases and the controls. And what you notice is that the pathways that are differentially engaged are all upregulated in the people who became infected compared to the people who remained uninfected. So we have significant differences between cases and controls at the pathway level and to a lesser extent at the individual gene level. So we could build several models, like I showed you before for the phenotypes, on pathways analysis in various cell subsets that showed a high accuracy of prediction of HIV acquisition. And the pathways in the myeloid dendritic cells contain those subsets with the strongest, those pathways, sorry, with the strongest predictive value at any visit. You can see here that's a very high accuracy and p-value. And these are some of the predictive pathways that we identified. In the myeloid dendritic cells, the so-called interferon gamma STAT1 signaling pathway is significantly elevated in those individuals who became infected. Inflammation. In the monocytes, type 1 interferon signaling. In CD4, T cells that express CCR5. Again, the interferon gamma STAT1 signaling pathway is upregulated. These pathways are upregulated. CD8 T cell memories pathways of activation as well. So you can see this in these examples. I'm going to show a few examples from in these heat plots. Increased inflammation in myeloid dendritic cells. These are the different time points over here, yellow, orange, and purple, showing month 0, 7, and 12. The people who remained uninfected in blue, the people who became infected in red, and you can see for the interferon gamma STAT1 signaling pathway, there's upregulation in those individuals who became infected at all time points. The same for the IL-1 processing pathway, another marker or another pathway of inflammation. In CD4 T cells, this activation and proliferation pathway upregulated in the people who became infected. And in monocytes, type 1 interferon STAT1 to IRF9 complex, the genes are here, activated in those people who became infected. So if we build the model, and I'm showing you here the model built on myeloid dendritic cells, what we find is that the model that we build at month 12 provides 83% accuracy prediction for HIV acquisition. This is extremely high. The pathway that contributes the most to this model is the interferon gamma STAT1 pathway in myeloid DCs. We can get the accuracy of prediction up to about 85% if we include pathways in other cell subsets as well, especially activation of CD4 T cells. So we should ask some questions. Is this gene expression profile stable? You might say, well, maybe those individuals got some infection, like flu, just before they became infected. And it's got nothing to do with some innate mechanisms within these individuals. Is it affected by vaccination? So what we did was a sample-level enrichment analysis to account for this. And what you can see, we're looking at the vaccine recipients in the filled circles, the placebos in the open circles, the people who remained uninfected in blue, the people who became infected in red. This STAT1 response pathway in mDCs is completely stable with time. So this is something that is in these people. And there is no effect of vaccination. This all overlaps. What we can also do now is to take these three types of measurements that we've been doing, the soluble analytes, the cell surface phenotypes, and the transcriptomics, and analyze them all together using a technique called mixomics. What that does is a progression-based approach that converges different types of data sets, different units, and different scales on a single uniform platform. So the input was the analytes from the ELISA and Luminex data, the cell subsets from our FACS analysis, and the myeloid dendritic cells with our transcriptomic analysis. The outcome was that we found an association between the frequencies of activated effector T cells, levels of soluble plasma biomarkers, and the STAT1 response pathway in mDCs to build a model with about 87% accuracy. The interesting thing here is if you put all of this together into the model, then you pull out one of the plasma analytes, which remember, none of them could predict anything when we analyze them on their own. But if you do mixomics, then soluble CD14, which is a marker of LPS activation of macrophages in vivo, that gets pulled into this model. So weak and strong markers can be combined for robust model predicting HIV acquisition. So that's where we got to a few months ago, and we thought, okay, this is very nice. Let's start looking at these pathways, looking at the genes, and trying to understand how they are responsible for acquisition or not. But something interrupted us, which was we remembered, of course, that we did the whole transcriptome, the gene expression analysis by whole transcriptome sequencing, right? So we had sequences, and we thought, well, let's look at the sequences, and let's see if we can find polymorphisms, SNPs, insertions and deletions, indels, that are associated with HIV acquisition. So I'm showing you how we go about doing this now for the indels. Indels are insertions and deletions. So from the RNA sequencing data of all these subsets, you can see that we get hundreds of thousands of indels that we identified. And what we have to do is to find out which of these indels, or which of these SNPs are significantly associated with becoming infected or not becoming infected. So we do a filtering. We find which of these indels you find only in the people who became infected, or only in the people who became uninfected. Those are the significant ones. And the ones which are equally distributed between the people who became infected, people who remained not infected, we throw those out as the non-significant indels. And so we end up with a bunch of SNPs which can differentiate significantly between infection state. And then we go through this filtering exercise again and again and again until we hone down on a bunch of SNPs. And then finally, we do a final stage of filtering where we look for the indels that are significantly identified across the different cell subsets. And we ended up with genes, with indels and SNPs that could significantly differentiate between cases and controls. So this preliminary analysis suggested that we'd identified polymorphisms that contributed to protection from or susceptibility to HIV acquisition. So this is a little bit of a kind of circular slide, but it's a nice way of showing how these alleles segregate. So I'm calling them alleles now, all right? Protective alleles and susceptibility alleles. So this is the frequency of the protective alleles in the uninfected people, right? They're all up there. And this is the frequency of the susceptibility alleles in the people who became infected. At this level, and I'm saying at this level because looking for SNPs in your genome by whole transcriptome analysis, whole transcriptome sequencing, is not the ideal way of doing it because you have an imbalance in the expression of genes. But I'll come to that in a second. But so far, we've identified 3,000 alleles which are protective and about 3,000 alleles which make you susceptible to infection. Now, each allele on its own is significant, okay? However, it's not significant if you do a multiple comparison, Bonferroni correction, all right? But that doesn't matter. We're interested in the whole package of these alleles. We're going to deal with Bonferroni in a second, all right? If you take the whole package of alleles, you can very, very significantly, with a p-value of 10 to the minus 16, differentiate between those who are protected and those who are susceptible. Now, interestingly, we had made available to us all the single nucleotide polymorphisms, all the SNPs from the MAX cohort that significantly differentiate progression rate in people who are already infected, right? Those who progress rapidly and those who progress slowly. And we compared our SNPs with those SNPs, and there was only 7% overlap, almost no overlap, which tells you that the alleles, the SNPs that affect acquisition, are completely different from the alleles, from the SNPs that affect HIV disease progression once you're infected. So what are these genes? Well, I'm not going to name names now, and you're going to understand why in a second. We have a lot of them. A lot of these genes, these are some of the protective alleles. These are the top hits. Some of these are the ones in infected people, the susceptibility ones. A lot of them are genes that are known to play a role in inflammation, and a lot of them are genes that are known to interact with the HIV replicative cycle. CCR5 was not in there, and HLA class 1 was not in there. What I'm showing you here is the frequency of these protective alleles in the HVTN 505 sub-study in our cohort that we studied, compared to the frequency of these alleles in the 1000 Genomes project, all right? This is a project where they sequence the hell out of, it's now 2,000 people that they've sequenced, and we have all those data. So we can compare the frequency of our protective alleles in our cohort with the frequency of the protective alleles in these 2,000 European genomes. We used the European genomes because people in HVTN 505 were mostly Caucasian. So the protective alleles are at a very high frequency in the people who remained uninfected in our cohort. They're at a very low frequency in the people who became infected in our cohort. They are enriched compared to what we can call the general population in the people who remained uninfected, and they are at a much lower frequency in our people who became infected compared to the general population. Now, watch this. If we combine the infected with the uninfected, so we're looking at the whole cohort of our sub-study, all of the people that we looked at, there's a significant enrichment for protective alleles in HVTN 505 sub-study compared to the general population with a p-value of 1.5 times 10 to the minus 11, all right? So the vaccine cohort that we studied was significantly enriched for protective alleles. Let's do the opposite now and look at the susceptibility alleles. The people who became infected had much greater frequency of susceptibility alleles compared to the people who became uninfected. We understand that. That's how we chose those alleles. But if we look at the whole cohort together, there was no significant enrichment of susceptibility alleles in our cohort compared to the general population. So let me just summarize the polymorphism analysis now before I summarize the whole story. So what we found are protective and susceptibility alleles that can distinguish uninfected from infected sub-study subjects in the HVTN 505 cohort. And there is a significant enrichment in HVTN 505 sub-study for subjects who have protective alleles that protect from acquisition of HIV compared to the general population of uninfected Caucasians. The implication is that the outcome of HVTN 505 vaccine trial was predetermined by the host genetics of the cohort due to the recruitment characteristics, because the people that they were trying to recruit for this study were uninfected, high-risk men who have sex with men, all right? They were high risk for many years before they were recruited, and they remained uninfected. And it would make sense that you might select for people who are protected. What are we going to do next? This is where we are now. We're doing whole genome sequencing of all 83 participants in our cohort to confirm the alleles. Remember I said transcriptome sequencing is not great for looking at SNPs in your genome, so we have to get balanced genomes now. Whole genome sequencing on all 83 people, and we've done that. We finished that 2 weeks ago, and we're analyzing it. Once we've done that, we're going to down select on the number of alleles. We're going to know which alleles are true, which are sequencing errors, and so forth. And we're going to then do targeted sequencing of those alleles in the remaining 2,000-odd HVTN 505 subjects who are mostly HIV negative, right? We'd say most of those would probably contain protective alleles. And another 2,000 HIV positive people that we're going to get those samples from Drs. Lederman and Shaka. And we're going to do that to achieve a GWAS level significance for individual polymorphisms. And then we can ask, well, which alleles or combination of which alleles confer most protection? What is the function of these genes and these alleles? And can we merge those data with our pathways analysis, remember, which gave us a very good predictive accuracy? We can ask another question which is interesting. We can ask, well, in all the people who were susceptible according to their alleles, maybe there was a vaccine effect that we just couldn't detect before in HVTN 505. And we can confirm the findings in other vaccine trial cohorts, such as Pambili, which was the same vaccine tested in women in South Africa, the STEP study, RV144, which did not select for a very at-risk population in terms of the participants, and the WISE study, which is a natural history study in women. All right, let's summarize everything. Plasma analytes alone provide no predictive model for HIV acquisition. Frequencies of cell subsets give moderate accuracy of prediction, with activated HLA-DR positive CD4 memory T cells being the strongest differentiator between becoming and not becoming infected. RNA-seq transcriptome data from sorted cell subsets gives accurate prediction models. There are several inflammation-related pathways which are associated with increased HIV acquisition, higher inflammation in participants who subsequently acquired HIV. And the so-called interferon gamma response STAT1 pathway in mDCs is one of the most accurate predictors for HIV acquisition. Host gene polymorphisms may protect from HIV acquisition. And this raises the question whether we should reassess how we recruit participants for our HIV vaccine trials. So, systems biology is a fishing expedition. And as I often tell people, it's not a bad thing to go fishing, all right? Because sometimes, not always, but sometimes when you go fishing, you catch fish, okay? So I'd like to thank the people who have been and are still involved in this study. This is almost all done by Christelle, an amazing postdoc in my lab. The bioinformatics was done by Jan Fay in my lab and Slim in Rafiq's lab. I have to thank the HVTN 505 team who've been amazing, especially Nicole Fram. These people who give us the monies to do the work, because this is probably the single most expensive experiment I've done in my life. And I'd like to thank you all for listening. Thank you.